Okay, uh, today we will see the question number 1 from biology paper 5090, paper 42, variant 2 of the October-November 2024. So this is alternative practical. Question is that carbohydrates are found in different plant organs in the form of starch and sugars, such as glucose and maltose. A student decided to test two different plant organs, potato and apple, for the presence of starch and sugar. Both of these are carbohydrate. Starch is the polysaccharide, and this is the storage form of glucose. And sugar, here sugar means glucose or maltose. In chemistry, these are called the reducing sugars. Anyway, so here in biology, the question is very simple. Suggest a suitable reagent for the test of starch and describe how it would be used. Detail of the possible results are not required here, and it has 2 marks. So actually, whatever you have learned in your theory, that starch is detected uh, by the reagent, that is iodine solution. And if a starch is there, then iodine make a complex with the starch molecules that is gray blue in color. So how will you write this answer? That the given sample is cut and crushed, because they ask about how it is tested. So if you are provided the plant organs of apple and potato, so from these organs, what we do? We first cut and crush the sample. Then a few drops of iodine solution is poured through a dropper. So the reagent is iodine solution, and this is the way how can we test it. Now, the student carried out the test for starch on pieces of the potato and the apple. Figure 1.1 shows, this is the figure 1.1, shows the appearance of plants before it was like this, that there is no color, and after the test. After the test, it shows that there are some patches of the actually, this is gray black in color, gray blue color, and this is also gray blue color. And sometimes it appears black if there is too much starch. So it is very clear that it has too much starch, and here apple has little starch. Record in table 1.1 what you have observed in figure 1.1. So see what is the table 1.1. It shows the two plant organ, apple and potato, observation and conclusion. So as I have told you that observation is that sample turns partially black in case of apple, and sample turns fully black uh, in case of potato. So why it happens that there is a small quantity of starch in apple, but potato contains large quantity of starch. So in this way, we have to write our answer in short phrase in the given table. Now, B part, the student then carried out a test for sugar on the potato and the apple using Benedict's solution. Now they have mentioned the reagent. Cut 1 centimeter, 1 centimeter, 1 centimeter cube of potatoes. You can say that is the 1 centimeter, 1 cm cube of the potato. On a white tile, use a cutting device to cut the tube into small pieces and place these in a large tube. Actually, when you have cut these tubes, and after cutting this tube, suppose this is a 1 centimeter cube, and now you have put these cubes in a large test tube. Then we have added 4 centimeter cube of distilled water and use a stirring rod to gently crush the pieces and mix them with the water. Crushing and mixing with water. So crushing so that sugar can be extracted out and dissolve in water. Now clean your tile, cutting device and stirring rod. So this is the normal um, procedure that you should keep everything clean around you and clean the tabletop as well, or whatever the tools you are using after completing your experiment and during the experiment as well. Repeat this procedure with the apple. Means here we have done this with potato, and now you do the same procedure with apple. So in this way, in two test tubes, or you may use more than two test tubes, you can uh, repeat the experiment in another test tube. So anyway, so then we add 8 centimeter cube of Benedict solution in both the test tube, means the test tube containing potato sample and the test tube containing apple sample. The contents of both test tube will be blue because Benedict solution is blue, so it shows is blue color. And as you also study chemistry, then remember that Benedict solution contain Cu2 plus ions, and this blue color is due to the Cu2 plus ions. Now heat the two test tubes for 10 minutes and a temperature between 75 degrees Celsius to 85 degrees Celsius. That is the standard temperature for this test, or you can say the suitable temperature for this test, Benedict's reaction. Now, describe how you would keep the test tube at the required temperature for 10 minutes. So actually, when you are instructed to keep the temperature between 75 degrees Celsius to 85 degrees Celsius. So there should be a water bath with the thermostat that keep the temperature in this range. So we can keep the test tube at the required temperature by using thermostatically controlled water bath. Now see, next. After 10 minutes, the student observed the test tube. The color of the apple mixture was red. So you should remember that when we perform Benedict test, then we can observe any of these color. It could be green, it could be yellow, or it could be red or orange. So actually green color shows low concentration of glucose or the reducing sugar, and yellow color solution shows comparatively higher concentration, and red is showing the higher concentration of sugar. While in case of potato, the mixture was blue, or you can say mixture remains blue. It means there was no reaction. So instead, what conclusion can be made from these results? So definitely, if it was red, so we can say that apple contain reducing sugar, or we can avoid the word reducing because this word is not included in our syllabus of biology. So you just say that sugar is present in an apple and sugar is not present in potato. Now, suggest why it was important to use 1 centimeter cube for each plant organ. Definitely, when you perform uh, a comparative test between two samples, then there should be everything should be same. The amount of reagent that you are taking, it should be same. The temperature should be same, and the duration when you are heating, it should be same. So that we can compare the results of the different samples. So this is the reason. Cut each tube up into small pieces to increase the surface area and to release sugar. I have already told you when I was discussing the description of the test, description of the procedure. Then crush the small pieces and mix them with water. So why it happens? So that sugar can be released into solution or water. So actually the main reason for this was the surface area, to increase the surface area. And during this, some of the sugar will be released. But when you will crush it and mix it with water, then definitely it will release and it makes a solution in water. So the last part of the question is that explain why the tile, cutting device, and string rod were cleaned after using them on the potato. So remember that we have to. Uh, there are two reasons. One is that your tools and the tabletop must be clean whenever you have finished your work. And secondly, here they are asking after using them on potato and after using them potato, what we have done. We have done the same procedure with apple. So if we don't clean the tile or cutting devices, so there is a chance of mixing of the contents of the potato over there with the apple. So our result would not be it was not accurate, and it may be contaminated with the sample of the potato. So that's why we want to prevent transfer of potato or whatever is starch or sugar, if anything is there in potato, uh, into the sample of apple. So that was the last part of the question. Now this is the C part of the question, that describe how you would test the apple for proteins and instead what you would observe if protein was present. For protein, we conduct biuret test. In biuret test, first we take the sample, and after the sample solution, same quantity of sodium hydroxide is added, and then a few drops of copper sulfate is added again. So then we shake the tube. Then after some time, a violet or purple color appears. So cut the apple with a knife, then crush the pieces, these pieces by the glass rod. So then crush those pieces which you have cut and crushed by the glass rod. And then we will prepare the solution. And after making solution, we will add sodium hydroxide and then 1% copper sulfate solution. And then we will shake it. So the solution turns from blue to purple, lilac, or violet. These are the different shades of the violet. If protein is not present, then it would remain blue.